begin the presentation. So I would also like to begin by acknowledging the Bunurong and Wurundjeri people of the Kulin Nation, who of course are the traditional custodians of the land, water and wildlife in the Melbourne region. Now, most of us have been working from home for some months now, so I thought it might actually be nice to transport you momentarily to somewhere a little bit more exotic. So I want to begin by telling you a story, and it's a story that's set in Mexico in the eastern Sierra Madre, which is home to a group of fish known as swordtails. Now, several species of swordtail fish actually occur in the same river system, and here I have an example of two such species. So we have a picture of two males here interacting with each other. The male down the bottom here is a male Xiphophorus malinche, and you can see that it bears the elegant long sword-like ornament that gives males of this particular group of fish their common name, the swordtails. And up above, we have a male of a different species, Xiphophorus birchmani, which has a much more modest sword ornament. It's also more high-bodied, it has these distinct bars, and has a much larger dorsal fin. Now, one tributary where these two species are found is the Rio Caunali in Mexico. And unfortunately, the Rio Caunali is subject to a lot of the human kind of impacts that we see in waterways right across the world. And what's interesting about the Rio Caunali in the context of swordtails is that in the 1990s, researchers did a comprehensive survey of swordtail fishes right across Mesoamerica. And when they came to the Rio Caunali, they reported these two species of swordtail fish living side by side. But less than a decade later, in the early 2000s, researchers came back to this particular river and they found that the two original species of swordtails have been replaced by a hybrid swarm. So here I have a picture of some of the hybrid males that are collected from the Rio Caunali, and they basically bear the traits that we find in the original parental species. So together with collaborators from the United States, we were interested in trying to understand what could be the underlying mechanisms that have led to these hybrids. Now, we already know from previous research that's been done on this particular group of fish that female swordtails show very strong preferences for the odour cues of males of their own species. And they rely on these odour cues to be able to make correct choices about whom they should be mating with. And so we collected Xiphophorus birchmani and Xiphophorus malinche from other rivers where they still existed. And we brought them back to a temporary research field station that we had set up by the Rio Caunali. And we tested the female swordtails for the odour cues of their own species versus the odour cues of the closely related sympatric species. So here are the results for Xiphophorus birchmani. And what we found was that, yes, in common with previous research, we found that female birchmanis showed a very strong preference for the odour cues of males of their own species. And they spent significantly more time hanging around with the source of that odour cue compared to the odour cues of the closely related Xiphophorus malinche. So we found this when we tested the females in spring water, and we also found this when we tested females in the treated municipal tap water. However, when we tested female birchmani for their responses in water that was drawn directly from the Rio Caunali itself, we find that this strong preference for the odour cues of their own males actually disappears. Now, the reason why I'm using this example is because I want to really sheet home the fact that behaviours, including reproductive behaviours, can have profound ecological impacts by changing the way different species interact with each other. And by disrupting the reproductive process and reproductive isolation, disturbances to the environment can also impact the course of evolution itself. So let's return back to Australia, to Victoria. And here I have an image taken of a stream in urban Melbourne. And it's actually close to a discharge point for treated wastewater. So there's a wastewater treatment plant located near this stream, and the treated effluent actually goes into the stream itself. So together with a postdoc of mine, Muna Saristo, who's now based at the EPA, we decided to look at the reproductive behaviours of another species of fish, in this case the eastern mosquitofish, which belongs to the same family as swordtails. We wanted to look at the reproductive behaviours of fish that were collected downstream from sewage treatment, and compared it to a more pristine reference site located close to a reservoir which was collecting water for human consumption. So a relatively pristine reference site versus a downstream from this sewage discharge. And I'm going to show you now the results just for the male mosquitofish. Again, we have association time, so the amount of time that male mosquitofish spent trying to get to the females, and we find that the males that were collected downstream from wastewater treatment site actually increased or had higher mating activity compared to those from the relatively pristine site. So again, we see a shift in behaviour, in this case between pristine site and downstream from wastewater treatment. Now you might think that an increase in mating activity is a good thing, but bear in mind, I did mention, of course, that mosquitofish are an invasive species in Australia. So an increase in mating effort is not necessarily a good thing. So it also suggests that there's something in the water that might be influencing the behaviour or disturbing or changing the behaviours of these animals. Now, we know that chemical pollution globally is an insidious form of environmental change. And in recent decades, concern has been mounting over pollution of the environment by pharmaceuticals. Now, everyone here would appreciate the therapeutic benefits of the medicines that we take or that we give to our pets or to our livestock. But what's not very well appreciated is that vast quantities of these drugs can actually end up in the environment. And the reason for this is because many of the medicines that we take and their metabolites actually remain biologically active when they're excreted. It's an unfortunate reality that most modern wastewater treatment processes, while they're very good at removing the solids and also the nutrients, they haven't been designed to remove pharmaceuticals before the water is eventually discharged. And as a result, they do enter the environment. And once these pharmaceuticals are in the environment, they're a cause for concern for three main reasons. First of all, many of these toxicants can be highly environmentally persistent. So one of the pharmaceuticals we've been looking at, Trenbolone, which is a veterinary pharmaceutical, has a half-life of some 265 days in the environment. So these drugs can persist for a very, very long time. Another problem is that the receptors that are targeted by many of these drugs tend to be evolutionarily conserved among different animal groups. So what that means is that drugs that might have been designed for you or I or for our livestock can also impact non-target organisms such as wildlife. And these effects can manifest at extremely low concentrations. So we're talking about exposures that are sometimes in the parts per trillion or nanograms per litre. Now, one infamous example of the ecological impacts of pharmaceutical contamination was sheeted home in a study published a few years ago, showing the demise of populations of vultures on the Indian subcontinent as a result of birds ingesting livestock carcasses that have been tainted by a veterinary pharmaceutical, a kind of anti-inflammatory drug. In the aquatic realm, we know that pharmaceuticals can also impact aquatic organisms. A paper published a couple of years ago, in fact, showed the presence of pharmaceuticals in the tissues of fish and invertebrates living adjacent to one of the research stations in Antarctica. So we're seeing these pharmaceuticals turn up in the environment and in wildlife, even in the most remote regions on the planet. Close to home here in Victoria, as Moira alluded to in her intro, study published by colleagues from the School of Chemistry a couple of years ago reported the presence of pharmaceuticals in waterways here in urban Melbourne. Some 70 different pharmaceuticals were detected in that particular study. And the important thing is that the pharmaceuticals weren't just turning up in the water, they were also found to bioconcentrate and bioaccumulate in aquatic organisms, including in invertebrates. And the researchers estimated that based on what platypus are able to eat during the course of a day. They estimated that platypus in some of these contaminated sites could be consuming up to half a human daily dose of antidepressants every single day. Which brings us to the antidepressant fluoxetine. So members of my research group and I have been studying fluoxetine, commonly marketed as Prozac, which the CSIRO has described as a drug of emerging concern. So it is one of the most widely prescribed antidepressants in the world, and it belongs to a class of drugs known as serotonin reuptake inhibitors, which, as the name suggests, act by inhibiting the reuptake of the neurotransmitter serotonin, which plays a really important and complex role across a whole range of vital physiological processes in the body. And of course, as a drug that's been designed to try and treat depression and also anxiety, it is of course a drug that specifically targets physiology and behaviour. And like many other pharmaceuticals, it's not completely removed from wastewater treatment before the effluent is discharged. And as a result, it has been detected in the environment in surface waters around the world at concentrations up to 929 nanograms per litre, including here in Australia. So fluoxetine has turned up in a chemical analysis of water taken from Sydney Harbour, and also in freshwater environments here in Melbourne. We also know that from previous studies that exposure to fluoxetine can have all kinds of sublethal effects. So studies done on Patermopurgus snails have shown that exposure to fluoxetine reduces the number of offspring that snails produce. Work carried out on Siamese fighting fish has shown that exposure to fluoxetine impacts aggressive behaviour. And a study done on the tadpoles of African clawed frogs have shown that exposure to fluoxetine can impair growth and development. So members of our group were interested in looking at how fluoxetine might potentially also impact ecologically important behaviours. And of course, some of the most important behaviours that can have a direct bearing on survival is simply being able to evade capture by would-be predators, such as this aquatic insect here. So together with a student, Jake Martin, we decided to look at the impacts of fluoxetine exposure on antipredator behaviours in the eastern mosquitofish. And we carried out a couple of experiments. The first one involved exposing mosquitofish to a predatory dragonfly nymph. And this next slide shows you a short video and also the setup. So we placed the fish into a tank that had grid squares marked on the bottom so that we can measure the activity of the fish either in the presence or absence of a dragonfly nymph. And here you can see a trial with the dragonfly nymph present. Okay, so turning to the results. So I'll talk you through this, this graph. So on the horizontal axis here, we have the three different treatments. So fish were either exposed to freshwater control, they were exposed to either a low concentration of fluoxetine for several weeks, or a high concentration of fluoxetine for several weeks prior to these tests. So these concentrations that we chose were environmentally relevant. They reflect concentrations that we find in the wild. And on the vertical axis, we have activity levels. So a high number means that fish were highly active. And here we see the results for when a predator was present, indicated by the maroon bars, and when the predator was absent, indicated by the white bars. And what we found was that irrespective of whether the predator was present or absent, fish that were exposed to low concentrations of fluoxetine were significantly more active compared to fish that were just exposed to fresh water. Interestingly, we didn't find any difference in our high fluoxetine exposed fish. So this is actually an example of what's known as a non-monotonic dose response, where we find an effect at the low concentration, but not at the high concentration. Now this result is important because many traditional toxicology and ecotoxicology studies tend to expose organisms to concentrations of toxicants such as pharmaceuticals at levels that far exceed, in some cases, many orders of magnitude higher than what we actually find in the environment. And what we are showing here is that exposing animals to environmentally relevant concentrations of pharmaceuticals matter. And more importantly, that oftentimes we see effects manifest at lower concentrations rather than at the higher concentrations. We also carried out an experiment where we simulated a predatory strike And using high-speed cameras, we're able to then look at the subsequent behaviours of the fish. And one of the behaviours we looked at was the amount of time that the fish spent freezing after the simulated predatory attack. Now, freezing behaviour is widely used by many species of fish and tadpoles to try and minimise the chances of being detected by the would-be predator. So again, we found that there was an effect of fluoxetine exposure on this important anti-predator response. But moreover, we found sex differences. So the impact of fluoxetine on freezing behaviour varied depending on whether it was females that were exposed or whether it was the males. So first of all, looking at the females, we find again a non-monotonic response. So females that were exposed to low fluoxetine spent less time freezing, which meant that they were spending more time active after the simulated predatory strike. By contrast, for males, irrespective of whether they were exposed to low or high concentrations of fluoxetine, males in this case, when exposed to fluoxetine, spent less time freezing and more time active. Now, so far I've been talking about the results of some experiments we've done looking at the impacts of fluoxetine, which is widely used to treat humans. But of course, it's important to bear in mind that it's not only human pharmaceuticals that are potentially problematic when they enter the environment. And I already showed that, this to be the case through the example of the plight of vultures on the Indian subcontinent following ingestion of these veterinary pharmaceuticals. So members of my group and I have also been looking at the impacts of a veterinary pharmaceutical, Trenbolone, and how Trenbolone might affect wildlife behaviours. So Trenbolone is an active ingredient in hormonal growth promotants that are administered to livestock, including beef cattle, to promote meat yields. Now, banned by the European Union since the 1980s, Trenbolone and hormonal growth promotants are still widely used in many parts of the world, including here in Australia, in parts of Asia, and in the Americas. So as a point of comparison, the United States produces some 20 million heads of cattle, representing 60 to 90% of the country's entire annual beef cattle production. They involve animals that have been treated with hormonal growth promotants. And closer to home here in Australia, some 2 million tonnes of beef, representing 40% of our beef cattle production annually, involves animals that have been treated with hormonal growth promotants. Now, Trenbolone is a powerful synthetic steroid with 15 to 50 times the androgenic and anabolic potency of testosterone. Those of you in the audience who are gym junkies or are into bodybuilding might be familiar with Trenbolone because it is one of these drugs that some people actually abuse in the gym to help them to bulk up. Now, in the context of cattle, Trenbolone is administered in the form of implants. Once inside the body, Trenbolone very rapidly breaks down into its most biologically active metabolite, 17-beta-Trenbolone, and it's this that actually enters the environment. So here we have a satellite image of some cattle feedlots taken in the United States, and you can see the liquid discharge from these feedlots are running into this pond in the middle here. So not surprisingly, like fluoxetine, Trenbolone has been detected in the environment, in this case up to 162 nanograms per litre worldwide. Now, earlier I actually used Trenbolone as an example of a pharmaceutical that's highly environmentally persistent with the half-life of 265 days. And chemists a few years ago published a study showing that the unique properties of this particular compound actually contributes to its high environmental persistence. So the researchers discovered that Trenbolone as a compound will actually break down when exposed to light, to UV, but it then has the ability to then reform again under the cover of darkness, leading the researchers to refer to Trenbolone as a vampire steroid or a zombie steroid. And we know from work that's been done previously that exposure of zebrafish to Trenbolone can lead to some rather alarming sublethal effects. In this case, complete female to male sex change or sex reversal. Now, given the role of sex hormones in reproduction, including reproductive behaviour such as the male courtship antics of this particular bird, rifle bird, we were interested in trying to understand how exposure to Trenbolone, an endocrine disruptor, might impact reproductive behaviours. But of course, we're interested in looking at this question not in birds, but rather in fish. This was work carried out by two other students, Pat Tompkins and Michael Bertram. And we decided to investigate this question in guppies. And the reason why we chose guppies is because they are a model system in behavioral and evolutionary biology. And it might surprise you to know that there are many, many papers published on the reproductive behaviours and reproductive antics of guppies. They're also popular aquarium fish, which makes them highly amenable to captive laboratory studies. So in guppies, males are more colourful compared to females. So males have all these different coloured blotches on their flanks. And we know that female guppies are highly choosy and discerning when it comes to their choice of mating partners. And one of the traits that females seem to find irresistibly sexy in male guppies is the amount of orange coloration on the male's flank. And a paper published in Nature a few years ago showed that the amount of orange is actually a reflection of the male's genetic quality. And so females prefer to mate with more orange males, and in so doing, they end up producing offspring which themselves are also more attractive to mate, so they can increase their reproductive fitness that way. And it's also been shown that by mating with males based on the amount of orange coloration, females can also influence the longevity of the offspring that she subsequently produces. So mate choice is very important in this species, and by being choosy, there are all kinds of population level repercussions in terms of the quality and quantity of offspring that can be produced. Now, this species is unusual in being internally fertilized. So males have got this modified fin called a gonopodium, which he actually uses to inseminate the female. And he can achieve copulation in one of two ways. He can either show off his bright colors and engage in a courtship dance known as a sigmoid display, or he can try and achieve copulation surreptitiously, essentially bypassing female mate choice by engaging in what evolutionary biologists call sneak copulation. So as the name suggests, the male sneaks up behind the female and attempts to insert his intromittent organ, his gonopodium, into the female's genital pore. So we're interested in looking at the impacts of Trenbolone exposure on both male and female behaviours. So first of all, I'm going to talk through some results for impacts on male behaviours. One of the experiments we did was simply to place Trenbolone exposed males and freshwater control males into a tank with a female, and we wanted to look at the competitiveness of the males. And what we found was that Trenbolone exposed males were aggressively more competitive compared to control males. Next, we carried out a separate set of experiments where we wanted to look at the male behaviours that were directed to females. So I'll talk you through this experiment. In this case, we presented a single male and placed him into a tank with a single female. And here you can see the different treatment combinations. So either a control freshwater male into a tank with a control freshwater female, a control freshwater male in a tank with a Trenbolone exposed female, Trenbolone exposed male in the tank with a control freshwater female, or a Trenbolone exposed male in a tank with a Trenbolone exposed female. So here we have the results for courtship, and what we found was that irrespective of the male's own exposure status, those that were paired with a Trenbolone exposed female spent significantly less time courting that female compared to control males that were placed into a tank with control females. And the reason for this was because those males actually switched their reproductive tactics. So they reduced their courtship and actually increased their sneak fertilizations, which, as you recall, is important because it actually bypasses female choice. So there are some implications of males switching this reproductive tactic because it bypasses female mate choice, which is very important. So what about female behaviours? Now, to address this question, we exposed or presented Trenbolone exposed females and also control females to potential suitors, presented one at a time over the course of two separate sessions or trials. So in the first session, a female was presented with a male. We then video filmed her behavioural response towards that male. We then, after that session was over, replaced that male with a second male, and we then repeated the whole process again. So we looked at the female behaviours when presented with two males, one at a time over 2 separate sessions. And recall earlier I mentioned that we know from previous studies that females prefer males that have more orange on their bodies. So we were able to directly manipulate the attractiveness of the males that we offered to females. So we offered females either high orange males or low orange males. So here we have the results for the freshwater or unexposed females. So in session 1 indicated by the dark teal and session 2 indicated by the light aqua. Here we have the different kinds of males that were presented to females. So in this case, it would be a low or unattractive orange male presented to the female in session 1, followed by another low orange male in session 2. Here would be a high orange male in session 1, followed by a high orange male in session 2, and so forth and so on. And again, we have association time, as a measure of mating preference and mating intentions. So first up, we found that control females were highly responsive. So over the course of the 15-minute trial, they spent 2/3 of their time, about 10 minutes, associating closely with the males. But importantly, I want to draw your attention to this particular treatment here, where we found that when females were presented with a high orange or attractive male in session 1, and then a low orange or less attractive male in session 2, she significantly reduced the amount of time that she subsequently spent with this less attractive low orange male. Now, the next graph I'm going to show you are the results for the Trenbolone exposed females, and it's on the same scale. And the first thing you'll notice is a dramatic drop in the overall sexual responsiveness of the females. So this time, an almost halving of the amount of time that females spent associating with the males. And importantly, the difference that we saw in this particular treatment in the control females disappears in the case of the Trenbolone exposed females. So these studies show that this powerful synthetic steroid, which is found in the environment, can affect both male and female reproductive behaviours, with implications for the quality and quantity of offspring that are subsequently produced. Okay, so where to from here? So I guess so far, a lot of the experiments I've spoken about in today's webinar have been short-term exposures carried out in the laboratory. So really the next steps in this field is to vary the scale and levels of ecological complexity, and we're already doing that. So in a world first here at Monash, we've been exposing guppies in large pools in greenhouses on campus to pollutants, pharmaceutical pollutants across multiple generations. So we're on to generation 15 now, so that we can study what the long-term impacts of pharmaceutical exposure might be on reproductive behaviours and also population dynamics. And thanks to the Future Fellowship, I'm also going to be doing some work with collaborators over in Europe once COVID restrictions ease. With these European researchers, what we're going to do is expose fish to pharmaceutical pollutants, fit them with tracking devices, and then release them into larger ponds and even into natural lake systems where we're able to use acoustic telemetry technology to study the behaviours of the fish in 3D in these lakes, and also monitor the fate, the survival of these animals under more naturalistic field conditions. So please stay tuned for the results in time to come. I want to finish off by thanking my various collaborators. Truly an international effort, this kind of research. And in particular, I want to acknowledge the people highlighted in pink, which are the students and postdocs who led some of the research I talked about today. And with that, thanks for your attention, and I'm happy to answer any questions.